Hi, this is Dr. Byson EM from Excellent Grades Academy. Today we're looking at muscle tissue, the histology of muscle tissue. We're also going to look at the gross characteristic and features of muscle tissue. Let's get into this class. All right. So what is a muscle? A muscle is a collection of muscle fibers. When we say muscle fibers, we mean myocytes. Myocytes are simply cells. So when we say myocytes, what we're simply saying, these are muscle cells. So a muscle is a collection of muscle fibers supported by connective tissue. Each muscle fiber is an elongated cell which contains contractile proteins, mainly actin and myosin. These two, actin and myosin, we're going to look at them when we zoom in the myofilaments that make up the muscle fibers. So interaction amongst contractile proteins causes contraction, which is a unique function of muscular tissue. Now, when we look at it embryologically, muscle tissue comes from the mesoderm of the embryo. So it is mesodermal in origin. Now, for you to understand muscle tissue, there are special terms that you have to understand for the organelles that are found in the muscle fibers. The plasma membrane is known as the sarcomere. The sarcomere. Then the cytoplasm is known as the sarcoplasm. The smooth muscle or the smooth endoplasmic reticulum is known as the sarcoplasmic reticulum. And this is specialized for storage of calcium in muscle tissue. The mitochondria is known as the sarcosomes. All right. There are three types of skeletal muscle. Let me just use a brighter color here. There are three types of skeletal muscle. There are three types of muscles rather. There is skeletal muscle. So it's already written here. Skeletal muscle. Skeletal muscle is also known as striated muscle because it has a lot of striations due to its arrangement of the light and dark bands that we're going to see when we start looking at the contractile units of the muscle, which is the sarcomere. This cardiac muscle. Cardiac muscle is specific to the heart. So this is muscle that is found in the heart. And we're going to look at the specifics and the characteristics of heart muscle. Lastly, but not the least, we've got smooth muscle, which is also termed as visceral muscle because it is found in most of the organs that are found in the body. Visceral muscle. All right, so apart from the above three types of muscles, there are also contractile cells that function as single cell units, namely the myoepithelial cells. Now, the myoepithelial cells, you may remember from a period when we were doing the glandular epithelia. So they are found in association with the cells that are found in the secretory acini of glands. We've got the myofibroblasts. These are involved in wound healing. So when we're doing wound healing in physiology, we looked at the myofibroblast. And we've got the myoid cells. These are found around the seminiferous tubules that are found in the testis, where spermatogenesis takes place. Let's zoom into these three types of muscles. Skeletal muscles. In skeletal muscles, the fibers are elongated and they are cylindrical. So they are like this. Fibers are elongated and they are cylindrical. So when you look at the embryology of how skeletal muscle is formed, you find that the myoblasts, which are embryonic cells that form the skeletal muscles, fuse. When myoblasts fuse, it's like multiple cells come together. So this myoblast and this myoblast and this myoblast will fuse. When they fuse, the skeletal muscle cell is going to be elongated and it is going to have a lot of nuclei. So they are multinucleated cells. And this nuclei that is found in skeletal muscle is found mostly beneath the sarcolemma. Now remember, we said that the sarcolemma is the cell membrane of muscle cells. So the multiple nucleus that are found in the skeletal muscle are found just beneath the sarcolemma. So this is a characteristic that is specific to skeletal muscle. They are multinucleated, they are elongated, and they are cylindrical. So this is what we're talking about. Skeletal muscle is also known as striated muscle. So these myoblasts are going to fuse and they are going to form a long cylindrical shape that is seen in skeletal muscle. And the nuclei are mostly located beneath the sarcolemma. The cytoplasm or the sarcoplasm of skeletal muscle is mostly filled with myofibrils. Myofibrils are simply myofilaments that make up the contractile units of skeletal muscles. Now, there's a very important cell that is found with skeletal muscles, and these are known as satellite cells. What is the function of satellite cells? Is the question. What is the function of satellite cells? So satellite cells function in regeneration of skeletal muscle. So satellite cells function in regeneration of skeletal muscle. They are very important. They function in regeneration of skeletal muscle. When skeletal muscle is damaged, satellite cells will function in regeneration. All right, so skeletal muscle fibers are supported by connective tissue framework, which can be well appreciated in cross-section of a muscle. We're going to look at the 3D diagram of how connective tissue surrounds the muscle tissue and individual muscle fibers. So the connective tissue framework carries blood vessels and nerves and also transmits the force of contraction through aponeurosis and tendons. The framework of connective tissue provides support to muscle fibers in the following manner. So we've got the epimysium, which is the dense connective tissue sheath surrounding the entire muscle tissue. And then we've got the perimysium, which is the connective tissue covering bundles of muscle fibers called fascicles. So there's an aggregate of muscle fibers, of muscle cells in the muscle tissue. And this aggregation of muscle cells in the muscle tissue is what we call fascicles. The fascicle is surrounded by a connective tissue that is known as the perimysium. The endomysium is a loose connective tissue that is composed of reticular fibers supporting individual muscle fibers. Let's look at the 3D diagram of this. Okay. So you can see what we see here. This is the muscle tissue. Okay. So the entire of this is muscle tissue. Let me just circle it. So the entire of this is muscle tissue. This connective tissue that you see surrounding the entire muscle tissue, this is what we call the perimysium. So that is the perimysium. And then in the muscle tissue, you can see fascicles. So there's a fascicle here. Okay, so the fascicle is surrounded by, so this was the epimysium, not perimysium, the epimysium. And then the fascicle is surrounded by a perimysium. And then an individual muscle cell, like that, is surrounded by an endomysium. So you have to know how connective tissue surrounds the muscle tissue, the fascicles in the muscle tissue, and the cells in the fascicles. This is a better diagram. Okay. So the connective tissue covering the entire muscle that is connecting to the bone here, that is known as the epimysium. And then the one that is surrounding the fascicle is the perimysium. The one that is surrounding the individual muscle fiber is what we call the endomysium. It's very important to know that. Okay. So this, we've already talked about this. This is the epimysium, connective tissue surrounding the entire muscle cell. And then the one that is surrounding the one that is surrounding the fascicles, that is the perimysium. Of course, this is just the bone here. And then this, what is attaching the muscle tissue to the bone, that is the tendon. And then this part that is covering the muscle, this part rather that is covering the muscle cell, this is the endomysium. Endomysium, depending on where you train from. Okay. So now let's look at the microscopic structure of a skeletal muscle. So each muscle fiber is an elongated, unbranched cylindrical cell. Elongated, it's elongated, it is unbranched cylindrical cell. It has many flat nuclei located just beneath the sarcolemma. So we've already looked at that characteristic. It shows cross striations. One thing that is very important about skeletal muscle is that it is striated. Okay? It shows cross striations of alternate dark and light bands with Z lines intersecting the I bands. So between the I bands, you've got Z lines. And between the Z lines, that is the contractile unit of a muscle that is called a sarcomere, something that we're going to look at very soon. So each muscle fiber is made of compactly packed long cylindrical myofibrils in the sarcoplasm arranged parallel to the long axis. Okay. So this is a skeletal muscle here. You can see that these fibers are elongated and they are unbranched. So this is a skeletal muscle here. The connective tissue that is surrounding each individual skeletal muscle is the endomysium. So this is the endomysium. And you can see dark, light striations. That's why it's called striated muscle, because it has a lot of striations. It has a lot of striations. Okay. Now let's look at the structure of a myofibril under an electron microscope. So the cross striations seen in the muscle fiber in the longitudinal under my light microscope is due to the presence of cross striations in the myofibrils. So each myofibril shows a dark, light and Z bands which are arranged in such a way as to give muscle fiber cross striations. So the distance between two Z lines is what we call a sarcomere. That is known as a sarcomere. The cross striations seen in a myofibril under the electron microscope is due to the presence of orderly arranged contractile protein filaments within it. Now, the arrangement of a sarcomere is that the sarcomere consists of two types of myofilaments arranged parallel to the long axis of the myofibril in a symmetrical fusion. So the filaments that are found in the sarcomere are the thick filaments, which are made up of myosin. Myosin, the protein called myosin, and these occupy the A band. And the thin filaments, which are composed mainly of a protein that is called actin. But there is also tropomyosin and troponin. So the thin filaments, you find three types of proteins there: actin, tropomyosin, and troponin. But in the thick filaments, you only find myosin. Okay. So as a result of this arrangement of the filaments, a band consists of thick filaments plus part of the overlapping thin filaments, and it shows a lighter zone in the center, which is called the H band. The structure of a sarcomere is uncompromising. You need to know this. There's no compromise when it comes to that. So let me use the diagram to illustrate. So a sarcomere is a region between two Z lines. So what you see here, this is a Z line here, and this is a Z line. Between two Z lines, that's what you call a sarcomere. The sarcomere is a contractile or the functional unit of a muscle cell. This line that you see in the middle here, this is what we call an M line, the middle line of a sarcomere. What forms the dark regions of the striations is this region here. That is what we call the A band. So the A band is formed by the regions that consist of the thick filaments and the overlap between thick filaments and thin filaments. So a region where you only find thick filaments in the A band, that's what we call an H zone. So the zone where in the A band you only find thick filaments, that's the H zone. The light filament is called the I band. So this from here up to here, that is called the I band. The I band, in the I band you only find light filaments, or thin filaments rather. Remember what we said. Thin filaments, there is actin, F-actin, there is tropomyosin, and there is troponin. So these are the types of proteins that are found in thin filaments. The three types of troponin. There's troponin T, C, and I. So that is what you find in the thin filaments. Thick filaments, the type of protein that you find there is myosin. All right. So contraction occurs when there is an interaction between myosin and actin. And then there's a sliding movement that is caused by ATP, and the sarcomere shortens. So when the sarcomere shortens, that's when contraction happens. So this is the sarcomere showing detailed structures. All right. So titin is a protein that will connect the thick filaments to the Z line. Okay, and the Z line is made up of a protein that is called desmin. So we've already discussed this. Okay, now let's look at the type of skeletal muscle fibers. From morphological, histochemical, and functional units points of use, the skeletal muscle fibers can be classified under three types. Namely, we've got the red, the white, and the intermediate type of skeletal fibers. The characteristic features of these three types of muscle fibers are due to the content of myoglobin. So this table here shows the differences between the red muscle fibers, which are also classified as type 1 skeletal muscle fibers, and the white muscle fibers, which are also classified as type 2 muscle fibers. The red muscle fibers have got high content of myoglobin and cytochrome. White muscle fibers have got low content of myoglobin and cytochrome. The white muscle fibers have got poor blood supply, whilst the red muscle fibers have got rich blood supply. The red muscle fibers are smaller in diameter, while the white muscle fibers are large. Mostly, the red muscle fibers are postural muscles, and the white muscle fibers are extraocular muscles. They are flight muscles in birds. Okay. The other thing that we need to understand are the motor end plates. The motor end plates are regions where the axons of nerves that innervate muscles meet muscles. That's where innervation of muscles occurs. The union between a nerve and the muscle, that is what we call a motor end plate. It is also known as a neuromuscular junction. So at the neuromuscular junction, this one here, at the neuromuscular junction, if there is a destruction or a reduction of a neurotransmitter is a chemical that causes the depolarization of a muscle cell at the neuromuscular junction. Then, if there is a reduction in that neuromuscular chemical, then you have a condition that is called myasthenia gravis. So myasthenia gravis is an autoimmune disease characterized by progressive muscular weakness that is caused by reduction in the number of functionally active acetylcholine receptors at the neuromuscular junction. So this reduction is caused by binding of the circulating antibodies to the acetylcholine receptors, thereby preventing the effective nerve muscle communication. So it is very important for you to know myasthenia gravis, and that it is mostly an autoimmune condition that is characterized by progressive muscular weakness due to a reduction in the number of functionally active acetylcholine receptors. So there is destruction in the acetylcholine receptors at the neuromuscular junction. That's how myasthenia gravis occurs. The other important aspect of skeletal muscles is knowing muscle spindles. So they are spindle-shaped encapsulated stretch receptors found in skeletal muscle, lying parallel to the long axis of the muscle. So each muscle spindle is made up of intrafusal muscle fibers and sensory nerve endings enclosed in a falciform connective tissue capsule. The intrafusal fibers are smaller in diameter than the extrafusal muscle fibers, which are the ordinary muscle fibers. The main function of the muscle spindle is to detect changes in the length of muscle fibers, which is just proprioceptive, a proprioceptive function. Now, Duchenne muscular dystrophy, which is DMD, is a hereditary disease of skeletal muscle, which usually affects males. So the disease is due to mutation of a gene responsible for formation of protein dystrophin on the inner surface of the sarcomere. The skeletal muscles become progressively weak from early childhood, and by adolescence the person becomes immobile. So Duchenne is a condition that is a must know. It must be known because it is a very high yield topic for exams. Let's move on and look at smooth muscles. So smooth muscles are elongated, spindle-shaped muscles that are mostly mononucleated. They are mononucleated. And smooth muscle is also known as visceral muscle because it is found mostly in the walls of viscera, in the GIT, in blood vessels, in the ureter, in the uterine tube, in the vas deferens, and a lot of viscera in the body. When you look at the histological picture of smooth muscle in viscera, this is how it occurs. So this is how it occurs. Mostly there are two bundles of smooth muscle. So you can see this bundle here. This is the longitudinal inner smooth muscle. And then this is an outer circular smooth muscle. So the characteristics of smooth muscle is that it is elongated, spindle-shaped. It has no striations. And it has a single nucleus. So these are the types of muscles here. So this muscle on top here, this is skeletal muscle because it is elongated, cylindrical, and it is multinucleated. Smooth muscle is here. You can see it is elongated, but it has a spindle shape. So spindle shaped, and it is mononucleated. Cardiac muscle is branched, as we are going to see as we go on. Okay. So cardiac muscle shows many structural and functional characteristics intermediate between those of skeletal muscle and smooth muscle. What we need to know about cardiac muscle is that it has numerous large T tubules, less abundant sarcoplasmic reticulum compared to skeletal muscle, and it contracts by the all or none principle, meaning that cardiac muscle contracts as a unit. Cardiac muscle is branched. So you can see there is a branch here. It is like this. Okay, so this is one cardiac muscle here. It is mostly mononucleated, and two cardiac muscles are connected by tight junctions that are known as desmosomes. These desmosomes are known as intercalated discs. So if this is cell 1 here, cell 2 is here, what is connecting them is this. This is a desmosome or an intercalated disc. Apart from intercalated discs, cardiac muscle has got gap junctions. These gap junctions function to transfer electrical impulses between two adjacent cardiac cells. So because they are gap junctions that specialize in conducting electrical impulses, cardiac muscle will function as a syncytium. What that means is that cardiac muscle will contract at the same time and relax at the same time. Cardiac muscle works together. That's why we said cardiac muscle functions by an all or none principle because of gap junctions. So these are the three different characteristics of these muscles. Skeletal muscle is striated. It is elongated, cylindrical, and multinucleated. It is voluntary. So you can control it at will. Apart from two skeletal muscles in the body that are not voluntary. They are skeletal muscle but are not voluntary, which is the diaphragm. The diaphragm is mostly skeletal muscle and it is involuntary. Also the cremasteric muscle, cremasteric muscle rather. Cremasteric muscle that is found in the testicles is skeletal muscle but it is involuntary. This is cardiac muscle. It is involuntary. You can't control it. It's found in the heart. It is branched and it is joined by intercalated discs. Further, it also has striations, but not as many striations as those that are found in skeletal muscle. Smooth muscle is long and cylindrical and it is spindle shaped. It is involuntary. You can't control it. It is found in the uterus. When contractions start, in regards to childbirth, you can't control them because they are made up of smooth muscle. The uterus is made up of smooth muscle. It is mononucleated and it has a very high regenerative capacity because it has a lot of satellite cells. Right. So these are the characteristics of skeletal muscle, cardiac muscle, smooth muscle. And you can pause the video here and look at them very quickly and see where the differences are between or among these three muscles. Okay, so in regards to regeneration, skeletal muscle shows a very limited regenerative capacity. So satellite cells living with skeletal muscle are implicated in the regeneration of skeletal muscles. Cardiac muscle lacks satellite cells and show very little degenerative or regenerative capacity beyond early childhood. When you damage your cardiac muscle cell, it is replaced by connective tissue. which will affect the contractility and relaxation capacity of the heart. Smooth muscle is capable of regeneration. It can undergo mitosis and replace damaged cell. So that's very important. Okay, that was a lecture. Now, self-assessment. Let's see if you've learned or gained value from this video. Answer these questions and score yourself. Write short notes on the sarcomere. What is the cardiac muscle? Differences between skeletal muscle and cardiac muscles. Then the neurotransmitter that is released at the motor end plate. What is it? The distance between the Z lines in a myofibril is called. The main contractile proteins present in muscle fibers are smooth endoplasmic reticulum of a muscle fiber is called. The diameter of cardiac muscle is about. And then these are MCQ questions. Choose the best answer. Which of the following muscle got good regenerative capacity? Which one amongst these options is the correct answer? So the cardiac muscle can be identified by the presence of that. Which one is correct? All right. This is Excellent Grades Academy. You can register with us for all recorded videos in anatomy, in histology, in DR sessions, gross anatomy, and in all the courses. WhatsApp us on +260 975 497790. I'll see you in the next video.